Stanford University.
We're very glad this is not a World Series night, if anyone cares. So let me just ask one question. Is there anybody here in the room tonight who has a cough or a fever? Because we're violating one of the most important rules of infection control. We're gathering together in this place. So let's hope we'll sustain ourselves. I hope you'll all be here next week. Now, I must say that in December, over the winter break, when I put together the course program and all the titles, as Kathy Gillum will remember, for this program and the ones that will follow, I did not at that point anticipate that we would be focusing tonight in part on H1N1 because that was December, and the whole issue about H1N1 really didn't happen until March or April of this year. And that really speaks to one of the dramatic changes that we're seeing, which is the emergence of global infections. In fact, some years ago, the Institute of Medicine of the National Academy of Sciences put forward a study that said that there could be a change in longevity in the United States and indeed worldwide for two reasons. One, obesity and all the consequences related thereto. And the second, the emergence of global infections. And we are, as you know, at a time when organisms emerging in one part of the world, and this is particularly true of flu, which does it every year, can migrate to others. And so this is an important saga. When we think about epidemics, we, of course, think about flu and we think about 1918, but there are many other epidemics that have taken place. And I mentioned cholera to you last week, and you can think about plague and so many other factors as well. But tonight we're going to focus on pandemics with a viral twist. Last week we focused on bacteria, and our speaker tonight, interestingly, Lucy Tompkins, is a world expert on bacterial infections. In fact, her research focuses on a GI organism that Julie Theriault did not speak about last week, which is an organism called Helicobacter pylori, which is a very fascinating organism because it is associated with cancer. And this is one of the interplays between microorganisms and a risk for cancer. But despite the fact that she is an expert in this area, she's going to put on another hat tonight. She actually wears several here at Stanford, in that she has been until recently the head of infectious diseases in the Department of Medicine. She is an associate dean of academic affairs in the School of Medicine. Tonight she'll not be talking about faculty challenges in behavior. But she's also the director of hospital epidemiology, and that puts her right at the epicenter of influenza and other hospital or community-related infections. And she was there from the very beginning of this event. I remember communicating with her over the weekend when the first description of H1N1 took place in Mexico, just to check in and see what was going on. And she was quite clear that they had been working all weekend on this problem. Dr. Tompkins is someone who's well poised to deal with these problems. She's both an MD and a PhD. Her MD comes from Dartmouth, her PhD from Georgetown. She's focused, as I mentioned, on bacterial pathogenesis. I've had the privilege of knowing her way before I came to Stanford because she has been a national and international figure in infectious diseases for quite a long time, and we had the privilege for one short period to serve together on the board of directors of the Infectious Disease Society of America, a testament to her leadership as well. So I'm really pleased tonight to welcome Dr. Tompkins to take us through the journey of pandemic influenza in this century and the last and beyond.
Well, Phil, there's one thing you don't have to worry about, and that's the obesity problem. Flu could get us, but... well, I'm not a virologist, as Phil said, but I've been fascinated by influenza for a long time, and actually one of my other jobs in the past was to serve on the National Advisory Committee on Immunizations for the Centers for Disease Control, where I finally got immersed in what it takes to produce a vaccine and how we should recommend vaccines for our citizens. And as an infectious diseases person, one cannot ignore influenza, and this is the perfect year to be talking about pandemics. So here we go. Now, I just have to give you a brief overview about a virus. First of all, viruses are parasites, and they really require a host cell in order to multiply. So they're somewhere in between a completely inert organic particle and another kind of life form. They don't have any of their own cellular machinery, but they do have nucleic acid at their core, and they all have a protein coat. Many viruses also have a membrane that surrounds them. Now, the nucleic acid can be composed of either DNA molecules or RNA molecules, and in order for viruses to do their thing, first of all, they have to be able to enter cells, and they do this very uniquely through some kind of an attachment mechanism and then enter the host cell, where they then co-opt the cellular machinery to make more copies of the nucleic acid as well as the protein coat. Then, to be successful, all viruses have to somehow get out of the cell and then finally be transmitted to a susceptible host. And so this is just a generic description of a virus. I'm going quickly, I realize that, but I don't really want to focus on the nitty-gritty of viruses. I really want to talk about influenza virus. So this is just a very simplified overview of the life cycle of influenza virus. As you can see on the left, let me see if I can get the pointer to show up here. You can see that the virus has to attach, and then it gets taken up. And then the viral particles, which in the case of flu virus are RNA genes, then have to be copied. And at the same time, the proteins that are encoded by these genes have to be made, and they use the host machinery to do that. And then ultimately, they have to be enclosed in another membrane in order to exit the cell. Now, I have a video that I want to show you that I got off the internet just recently. I think it's very helpful to sort of understand this life cycle. We're very lucky. This is about 3.5 minutes.
A virus is a small infectious particle that hijacks the machinery and metabolism of our cells to grow and reproduce. Each viral particle, or virion, consists of nucleic acid, either DNA or RNA, in single or double-stranded form, enclosed by a capsid. In most viruses, the nucleocapsid is enclosed by an outer envelope. Influenza type A, an enveloped single-stranded RNA virus, has been widely studied and serves here to illustrate the life cycle of one common virus. Scientists believe that influenza virus is spread from person to person through exposure to large respiratory droplets, direct contact, or airborne dispersal. Infection takes place mainly in the respiratory tract. Infection begins with attachment of virus proteins to a receptor on the surface of the host cell. The virus is then taken in the cell by receptor-mediated endocytosis. As the virus enters the cell, the virus is internalized in a membrane-bound capture vesicle that carries the viral core. The vesicle is transported on microtubules inside the cell by host proteins called kinesins. During transport, the membrane of the vesicle fuses with the membrane of the virus, and the capsid undergoes uncoating. The viral core RNA and proteins are then released into the cytoplasm, where they are guided by host proteins to the nucleus of the host cell. At the nuclear membrane, the viral core uses host protein channels to enter. Inside the nucleus, cell machinery is utilized by the virus to replicate the viral genome and make messenger RNA, mRNA. Some viral mRNA exits the nucleus to exploit cellular ribosomes to direct synthesis of viral proteins. Viral proteins go back to the nucleus to associate with viral RNA. These nucleoproteins again leave the nucleus and use cellular processes to travel to the cell surface. Viral surface proteins are made and processed in the cytoplasm and also travel to the cell surface, where they combine with encapsulated nucleoproteins to form progeny viruses which depart from the cell by budding. The virion now goes on to infect other cells. At present, vaccines to stimulate the production of antibodies against the virus and stimulate host cellular responses to ingest the virus remain the best strategy for controlling certain viral diseases. But they are of limited effectiveness against rapidly mutating viruses and are ineffective against viruses that cause the common cold and AIDS.
Okay, now there are two really important things to say about influenza virus that I hope will explain why we're having a pandemic. The first one is that there are two very important proteins on the surface of this membrane of the influenza virus A. One is called the hemagglutinin, and this protein is essential for the attachment to a host cell receptor called sialic acid. The other important protein is this neuraminidase, which is actually an enzyme that actually is critical for releasing the viral particles from the host cell once it's budded out. And this is another little schematic that I thought was helpful in showing that as the RNA and the proteins near the surface of the host cell, they actually make these hemagglutinin and neuraminidase proteins, which are studded on the side of the host cell. And then the whole thing buds off and into a viral particle. But it's got to get released from this sialic acid receptor that's still on the host cell, and so that's what it uses the neuraminidase for. The neuraminidase enzyme actually cuts this bond here so that the particles are free. Now, our drug Tamiflu, oseltamivir, and the other neuraminidase inhibitor drugs work by actually inhibiting the activity of the neuraminidase enzyme on the virus. So they keep the virus from spreading to other cells. That's an important feature of Tamiflu. Sort of explains why it doesn't necessarily prevent infection, but it does attenuate the infection. Now, the way influenza viruses are named by type, such as type A, as you see here, or type B. We're going to talk about A exclusively today because they are the most complex and they're also the causes of pandemics. The second thing here is the city for which the initial isolate was made, from which the initial isolate was made, the date that it was obtained, and then I'm having trouble with the pointer, but then the types of the hemagglutinin protein, in this case it's an H1, and the type of neuraminidase, it's an N1. That's the convention. And as I said, there are really three types of influenza viruses, but C is really not much of a pathogen in humans, and B doesn't undergo the changes that A does, and B doesn't give rise to pandemics. And so we're going to talk about A exclusively now. It turns out that there are at least, I believe, somewhere between 11 and 15 different hemagglutinin proteins, which all differ by a substantial amount when it comes down to the amino acid composition. There are 5 neuraminidase proteins that are found in nature. And in humans, in the last 100 years, we have really only experienced infection by 3 different H types and 2 different N types. Now, the other critical feature about the influenza virus that this slide shows again is the fact that it's an RNA virus, not a DNA virus. And the problem with RNA viruses in general, and especially with influenza virus, is that the copying machinery of the cell isn't very faithful at copying the RNA molecules. And so mistakes are made. Different nucleotide bases are inserted as mutants or mutations. And the second feature that's very important to understand is that the genes of the influenza virus are separated. So there are 8 different strands of RNA that are inside each viral particle. It's called a segmented genome, unlike the bacteria that Julie talked about last week, which have essentially a single chromosome where all the genes are linked together. Well, what does this mean? Well, it explains, I think, the 2 phenomena that we deal with influenza. First of all, as you can see on this slide, I'm going to talk about antigenic drift, and what that means is that because the virus continues to evolve continuously in nature, in its animal hosts, and mistakes are made regularly in all of these genes, in the copying mechanisms. But for instance, if mistakes are made in the hemagglutinin gene, where's my pointer? Darn. My computer is so sensitive. See if we can get this to work better. Okay. Then it's possible to change some component of, say, the hemagglutinin gene such that it's slightly different. Not very different, but enough different that if we look at the original hemagglutinin protein, if one injected this strain into a human, you would make antibodies that would be very specific and might block the attachment right here. But I don't know why my computer is doing that. But on the other hand, a slight change could essentially disrupt this binding. And that's the reason why we have a new vaccine essentially every year, because this process of antigenic drift takes place continuously, and the strains that are circulating in the community, like our seasonal flu strains that we had 3 types of this last year, are continuously changing a little bit such that the vaccine won't be as protective as it was the year before. And so we have to keep up with it. That's why you need to get an annual flu shot, one of the only reasons. Now, it also turns out that if the...
Let's give up on that thing.
Give up on this thing? Great, thank you, Kathy. This is welcome. If a cell happens to be infected with a virus from a human, and a completely different influenza virus from a duck, let's say, and they're in the same cell, the fact that these are all independent genes, they're independent of one another, and if they're all mixed up in the same cell, it's possible to have a reassortment, and for this basically human virus to pick up, say, a completely new gene from the duck, and let's say it's the hemagglutinin gene. Well, if it's a duck hemagglutinin, none of the antibodies that would work before against the binding of the hemagglutinin will work now, because this protein is entirely new. And this is the substrate for the evolution of a pandemic strain, and this is essentially what has happened in 2009, and I'll get back to that much later. So antigenic drift is really a process of minor mutations in the RNA that code for either the hemagglutinin or the neuraminidase. And these 2 proteins are really critical because they are what we use to vaccinate people with. These are the major outer proteins that cause an immune response and are protective. You had a question?
Yeah, I think you differentiated and you said seasonal flu viruses versus H1N1.
Right.
What's the reason why that's differentiated? And is it possible that H1N1 then just becomes part of the seasonal flu virus?
The question was, what's the difference between H1N1 and the seasonal flu viruses? Could I get back to that? Because I really do hope to explain that. That is the crux of what I'm going to try to say, I think. Thank you for asking, though. And then antigenic shift is when we see a whole-scale new virus evolve that has either a new hemagglutinin protein of a different type or a new neuraminidase or both. And over the last 100 years, we've actually now had 4 new hemagglutinins and 3 new neuraminidase antigens that we have seen. And I must add that even if it's called H1, not all H1s are the same. Actually, there are variations between the different H1s as well. They're not as substantial as between an H1 and an H2 protein, but they are significant enough that, for instance, the H1 of the seasonal flu virus, which we did have circulating for the last 40 years, is really very different from the new H1 that has evolved in California and Mexico. So the other important thing to talk about is the natural reservoir for influenza viruses. And in fact, all of those different combinations of H's and N's actually occur in migratory water birds. So they are the natural reservoir for influenza viruses in nature. And, but they can infect domestic fowl, especially ducks, with their viruses. Now, you'll also notice here that, and those ducks can infect domestic poultry, and that is essentially what happened in Asia, actually starting in the 90s, but now in 2005 and so forth with H5N1, that presumably the H5N1 evolved from water birds into domestic ducks and then into chickens and other poultry that are in Southeast Asia predominantly. But you'll also notice that pigs and humans can share influenza viruses very readily. They're very similar as far as the virus is concerned. So that a human virus can infect pigs and vice versa. And also the pig serves as a nice misting vessel for poultry viruses that are in the poultry. Any questions? No. We're going to keep going. Oh, yes, one question.
So why is the hemagglutinin is used to bind to the cells?
Right. The hemagglutinin is used to bind to the cells.
So why is variation in that sufficient to prevent antibodies from binding, but still allows the viruses to bind to the cells?
Well, the antibodies are things that we make to protect ourselves from being invaded, but
They're both sort of glommed onto the same. I mean, the cell is using the hemagglutinin to bind to the virus is using the hemagglutinin to bind to the cell, but it's the same binding site that the antibodies are attaching to, right?
yes.
So why is it that changes in the hemagglutinin still allow the virus to bind to the cell, but sort of prevent antibodies?
Oh, the question is, if the hemagglutinin is still has been changed and our antibodies don't react to it, why does it still actually work perfectly well to get the virus into the cell? And that has to do with the nature of the receptor. And so the receptor is more forgiving, if you will. And so the hemagglutinin isn't that changed. And it also depends on where the receptors are. And I think that gets into a more complex question that we shouldn't talk about right now. But you're onto the physical chemistry, actually, of receptor binding, and I can't discuss it. Now, symptoms of influenza. How many of you have had flu in the course of your lives? Just about everybody. So how is it different from the common cold? Well, usually there's a high fever that's associated with influenza. Usually there are muscle aches. And certainly cough is a predominant feature, whereas the common cold, is more upper respiratory tract. And if you've had flu, you remember your joints ache. And the current strain is actually causing a significant amount of GI upset, both diarrhea and vomiting, not only in kids, but in a significant proportion of adults. But these are the symptoms. And the transmission, as shown in the video, one major mode of transmission is through these droplets that are expelled when people sneeze or cough. And so if there's anybody with a cough here, I hope you will leave. If you're in the infectious period. And then we inhale these droplets, and they land predominantly in the back of the nose. It's called the nasopharynx, and the throat. The other mode of transmission is essentially these droplets land on environmental surfaces, but they also land on your hands if you happen to have the virus. And therefore, washing hands is a very helpful thing to prevent inoculation. So basically, the mucous membranes of our respiratory tract, our mouths, our noses, and our eyes are all susceptible to entry of the virus into the system. So hand washing, hand gel are very important. So let me talk about pandemics for a minute, because this word is used very loosely, and I think it's confusing, or it has been confusing to a lot of people. And there is no perfect definition. But as an infectious diseases person and epidemiologist, mainly what we mean by a pandemic, and especially as it regards influenza, is that it's a widespread epidemic that basically goes to multiple regions in the world. And the World Health Organization had this very complex system of calling it a pandemic when it got to so many regions in the world. But the fact is, influenza is global. And if it's a widespread epidemic of a brand new strain that becomes global, which it will, that's a pandemic. The other critical feature of influenza pandemics is that it means the majority of the population doesn't have any immunity. So for our seasonal flu vaccine, over the course of years, a greater and greater number of people develop some immunity to the seasonal flu virus. But then if a brand new virus enters the population of the world, and 95% of the people in the world don't have any immunity, then it leads to a pandemic. And usually these move very quickly, almost explosively. And in the case of influenza, we're talking about the ability to go from human to human. So the bird is really not part of this cycle at all after it gets started. Now, Phil wanted me just to differentiate, we've also talked about cholera pandemics, and this is another organism that can eventually move around the world or to many parts of the world. And the plague essentially was pandemic in Europe in the 14th century and other times of history. But influenza is a human-to-human transmission. These others either require an environmental reservoir, like the cholera organism, or they're spread by insect vectors, like the flea in the case of plague. Flu basically doesn't have any social stigma attached to it, at least not seasonal flu or pandemic flu. But we can point to several examples where having tuberculosis in the late 19th century certainly was stigmatizing in the early part of the 20th century. And certainly HIV was very stigmatizing. So these are other pandemic organisms, if you will, but the consequences are different in our society. The first actually well-documented influenza pandemic occurred in 1580. Of course, we don't have any virus from that period of time, but the symptoms and the historians have, I think, clearly shown that this is probably a very ancient disease. And our last pandemic, until this year, was in 1968. Now, I just want to get back to this point about the immunity and the role of immunity in the pandemic. If we look at the left part of the slide, which is the disease incidence, and the yellow curve, let's say it's 2009. We're going to have an explosive outbreak of flu, which we are, to which none of the population is essentially immune. That's the blue line. But over the course of years, to answer your question, what we expect is that this will play out exactly the same way as our other pandemics have, which is that we'll have epidemics, but as time goes by, the amount of antibody, if you will, or immunity that's in the population will continue to grow as a result of more and more exposure. And these epidemics will be smaller and smaller and smaller, and at some point in time, which is very hard to predict, although at one time it was thought to be fairly predictable, I don't think it is. This is a perfect opportunity for a brand new virus to be very successful because a brand new virus, there won't be any immunity in the population. And so it'll be to the advantage of a virus like that to emerge when most of the population actually has antibodies to the preceding virus. And then the whole thing starts all over again. So it does depend on the level of immunity and this, if you will, the fitness of the virus as it continues throughout the ages. And I think it would be impossible to say, okay, we're going to have another pandemic in 20 years or 40 years. We don't know.
Lucy, maybe you want to comment on how that affects different age populations, because H1N1 right now is affecting the elderly as much, and part of explaining that.
Yeah, Dr. Pisa wanted me to comment about the effect of different ages of the population. As I will talk about a little bit later, for instance, we know right now that in this brand new strain, the H1N1, the swine H1N1 that's circulating now, people who are over the age of 60 appear to be relatively immune. And that's probably because they have seen a similar virus 60 years ago or more that the rest of the population hasn't seen. And it does illustrate this point. Thank you for bringing that out. So in the 20th century, we have had 3 pandemics, and I'm going to concentrate on the 1918 pandemic because I think there are lessons to be learned for the contemporary pandemic. And then I'm going to talk about 1976, when we didn't have a pandemic, but we thought we were going to. But just take a quick look at the mortality rates, and I think you can see why 1918 has been fascinating just from this fact that it's estimated between 50 and 100 million people in the world died of influenza in that year. And the population of the globe was 1.6 billion people as compared to 6 billion people now. So you could do the multiplication, and in the United States, at least 550,000 people died from influenza that year. The subsequent next 2 strains, and this is the one that's been predominantly circulating for the last 40 years, were much milder. So they didn't have the same degree of virulence. I also want to point out right now is that we had no idea what caused the influenza pandemic of 1918. We didn't have the ability to culture the virus at that time, and there were a lot of questions about whether this was a bacterial infection or it could be a filterable agent, as it was called at the time. So, on to 1918. First of all, 1918 is when yes, you have a question.
You mentioned that they weren't able to culture, but you also had on there that it was the H1N1. How did they know?
So if we couldn't culture it, how did we know? And I'll tell you how we know in a minute. It's a great question. Yes, and another question.
Regarding this virus mutation thing, you talked about how it mutates. It looks like that if I get the virus, if I sneeze out, whatever goes out, it's very likely it is already mutated. And you say we have every annual vaccination process. there are two questions. One is that where does this annual period come from? Is whatever I have, within few months, it will be different virus. So that's one question. Second question is, if it's mutating, and the mutation seems to be a pretty random process, how does the drug company know next year what's the virus?
Yeah. Well, let me answer your second question, was how do the drug companies know what vaccine to make? And the drug companies, first of all, although you may have a few mutant viruses that came out of you when you sneezed, let's say, just in the course of having had flu, most of those might have other mistakes made that make them not able to cause any infection. So, and because there's a balance between your immune system and the virus and so forth, and I'm talking at a global level, not at an individual person level, the virus tends to remain relatively the same. It'll have the same hemagglutinin types and the same neuraminidase types over a prolonged period of time until there's one of these episodes that occurs with this big shift. But to get to the point about how we make the vaccine, I was going to talk about that a little bit later, but I can say right now that what happens is that the World Health Organization has a meeting every year, usually in February, where in the meantime there have been many, many sentinel laboratories all around the world, including the Centers for Disease Control here in Atlanta, Georgia, that contribute their strains to this huge strain collection. And they continually keep track of what influenza viruses are circulating around the world. And from that collection, the World Health Organization and this group of people who meet there make a decision about what the new vaccine, the seasonal flu vaccine, will contain. It always contains one influenza B virus, which really, they don't really change that much. So B just stays B. It doesn't have all these different hemagglutinins and so forth. And usually 2 types of influenza A. And they'll be named by the city from whence they were derived. And that is what all the vaccine manufacturers will get those strains from the WHO, and they will all make the same seasonal vaccine, essentially. Every once in a while, a new virus or a new variant, usually a new variant, might evolve, and that actually happened just about 2006, I believe. We kind of missed the boat on a strain that came up after this decision was made. And if you've been looking at all the news reports about how we're making the vaccine, it takes months and months to make it because it's made in eggs. It's a very old technology. So once the commitment is made to make the vaccine, there's really no going back. And so it may occur that a variant occurs that the vaccine is not going to be as protective against as you would like. And so people might still get the flu, or an attenuated case of the flu because the vaccine doesn't quite cover the latest variant that's arrived. Yes?
Also mean that different parts of the world might be having different strains and they may need different type of vaccine.
And the question is, are different parts of the world having different strains? Yes and no. There is a predictable travel of influenza viruses around the world. Generally, they start first in China and move to the northern hemisphere, through Europe and then to North America, South America, as the world turns, if you will. So generally, this covers the entire globe. What's happening in China eventually will happen in California. And it's very seasonal as well, and I'd like to talk about that a little bit later. Thanks. So back to 1918. Well, 1918, some of you might know. None of you will remember 1918. Not even I will remember 1918. But my mother got the flu in 1918. Was the year that the U.S. entered World War I, the Great War. And Woodrow Wilson, who was the president, was bound and determined for the U.S. to help win the war at all costs. And I just wanted to read you one quote from Wilson. He was actually talking about the Liberty Loan Campaign. The Liberty Loan Campaign was a way to raise funds to support the American involvement in World War I. And basically, if you were patriotic, you needed to buy bonds, which were used to support the war effort. And there were many acts passed that were, I seem to be highly unethical and illegal these days, but one was the Sedition Act. If you were not patriotic enough, or you spoke out against the U.S. from joining the effort, you could be put in jail. In fact, you could go to jail for years for just speaking your mind. The Sedition Act was resurrected, essentially. It is essentially the same as what Lincoln had to do with rescinding habeas corpus. And there was a commission that was designed to keep track of who was patriotic and who wasn't and so forth. And including the patriotism, your evidence of patriotism was to sell these bonds. And so what he said is, to open a Liberty Loan drive, Wilson demanded force to the utmost, force without stint or limit, the righteous and triumphant force which will make right the law of the world and cast every selfish dominion down in the dust. Does this guy sound like a preacher or what? So he had an incredibly religious zeal about this. And in order for the U.S. to help win the war, we had to mobilize, he intended to mobilize hundreds of thousands of recruits to the armed forces. And therefore, to do this, hundreds of cantonments were built to house thousands of men, either on their way over to the front or on their way home from the front. And these were built very hastily and were extremely crowded. There were thousands, sometimes 10,000 men in a single cantonment. All these were basically bases that were outside major cities, and for the most part. They were the perfect breeding ground for a human-to-human transmitted virus like influenza. This is an example of the sleeping quarters in the San Francisco Naval Training Station. And you can see the only thing they did here to try to prevent spread of an infectious virus was they put them head to toe. But if you can imagine 1,000 people sleeping in the same room, that's what was going on. And he was given advice by Gorgas, who was then the Surgeon General of the United States, and by Dr. Welsh and other experts that, these facilities were inadequate and improper, and were a catastrophe waiting to happen, but it didn't matter because we were going to win the war at all costs. This is an example of the Liberty Loan Parade that took place in Philadelphia right at the height of influenza in 1918. And you can see all the thousands of people who were gathered here to participate in the parade. Here's a cop who's wearing a mask. A lot of good that did. But you can see he's got his stop sign also has "Buy Liberty Bonds." So policemen were expected to do their patriotic bit as well. So flu began in the spring. It went to Europe probably from the United States. That's the best guess. It affected the troops in Europe massively, the Germans and the French, for instance, fighting on the Western Front. And it probably finally put the kibosh on the German offensive to try to win the war in June of 1918, in that so many troops were ill with flu that they simply couldn't do it. This is an example of one of these cantonments that's now been made into a hospital for the thousands of recruits who were ill with influenza. It's pretty amazing if you think about what it would take to take care of these people when we had really nothing specific to offer. I believe these are ill patients, but there were attempts to try to stop the spread of influenza, whatever was the cause, by masks. Mostly they were gauze masks, which we know wouldn't really prevent droplets from being inhaled. And in some places they used curtains. Now, Philadelphia was basically, I think, the hardest hit city in the United States. Essentially, flu came back with a vengeance from Europe, through the troops probably, moving back and debarking at the port of Boston. Boston was very hard hit, but shortly thereafter, with the troop movements, the cantonments outside of Philadelphia became affected, and then the population of Philadelphia became affected. They had so many deaths in such a short period of time in Philadelphia that they had a shortage of coffins, they had a shortage of undertakers, and they essentially had to go to burying people in mass graves. Eventually they had to use backhoes to dig the graves because they didn't have enough people to dig them. And the morgue in Philadelphia is shown at the bottom here. The bodies were just stacked up like cordwood. In fact, in Philadelphia, there were bodies outside the morgue on the street. And in other parts of Philadelphia, as well as other cities, there were people who were found dead in their homes. I met who'd been dead for days. And I met someone on a plane recently who told me he was an orphan. Both his parents died in the 1918 pandemic when he was about 2. He was adopted by his aunt. He didn't know he was adopted. Turns out he grew up in the same city where his brothers and sisters lived, and he only found out last year that he had brothers and sisters. This is not a rare story. We ran out of hospital beds. We didn't have nearly enough hospital beds to take care of the civilian population. So this is a hospital outside of Lawrence, Mass., which we used tents to take care of the civilians. We also had a tremendous shortage of physicians as well as nurses, but predominantly physicians because they were all called up to take care of the soldiers. And the Red Cross provided the nurses as many as they could find, and they were really much more important than the physicians since there were really no specific remedies, and nursing care was really the most important thing for resolution. In Philadelphia, they asked the medical students from the medical colleges there to essentially start taking care of patients as nurses. And so this is just a group of medical students all wearing masks who are waiting to be assigned to their duties. As I said, they didn't really know what the cause was, but spittoons were rampant in society then. Spitting was very common, and so they thought that spitting would probably not be a good thing, and so there were signs everywhere, and they took spittoons away. I think that was the beginning of the end of spittoons, except in Congress, where they still had them in the 40s. And there was some thought that clean, fresh air would be helpful. Probably not a bad idea, but probably didn't really work. And even the street sweepers wore masks. Now, this is the kind of data that we now see every year that's produced by the CDC, and it looks at the pneumonia and the influenza mortalities across the United States for every week of the year. And for purposes of epidemiologic statistics, it's defined that there's a seasonal predictability to when we have flu, and that there's a baseline expected number of cases of flu, depending on which week of the year it is. This is the winter month. And the jagged line are the actual cases, And so when the jagged line goes above what's predicted, it's considered to be an epidemic of flu. That's just the definition of an epidemic. So you can see here's the epidemic period, and it's 8% of all deaths are due to pneumonia and influenza. This is what it looked like for 1918. There are actually two curves here. I know you can't see this, but this tall curve are all deaths that occurred over this very short period of time. I think this is a matter of days. And this peak are deaths just due to influenza. So this is a massive, explosive epidemic with a massive number of deaths over a very short period of time. Two pictures tell it all. These are the death records for the United States in 1918 and 1919. And they're about 5 times as thick. Up to 5 to 10 times as thick as the average death record for a year would be. There was something else very strange besides the fact that so many people died, many more than would normally occur in a usual year of influenza. 10 times, at least 10 times as many people in the United States. The death rate may have been overall 10 times as great as it would ordinarily be. But there was something funny also about the age range of the people who died. And these are data from 1911 to 1917 in this line. And this is this is what we see virtually every year of who dies with influenza. It's very young people and very elderly people, or somewhat elderly people. I'm not using the word elderly anymore. It's amazing what happens when you forget it, when you hit a certain age, yeah. But look at what happened in 1918. Again, the young people, it's a much higher rate, but then there's this big peak right here between essentially 20 and 40, or 15 and 45. In fact, in the United States, 50% of the people who died were in this age range. What would you expect would happen to our actuarial statistics? Well, this is what happened. In 1 year, we lost 15 years of life expectancy because the most productive members of society died. And this is really what happened in Africa with HIV. The difference is that we recovered, obviously, because this wasn't a continuing epidemic. But now it looks like this in Africa. Now, as I said, there were a lot of theories, and in fact, the leading theory was that influenza in 1918 was caused by a bacterial bug called Pfeiffer's bacillus, now which we call Haemophilus influenzae. And in truth, a substantial number of the people who died of influenza actually died of the bacterial superinfections that occurred, primarily pneumonia, following the onset of influenza. But in 1930, Robert Shope isolated the influenza virus from swine for the first time and showed that indeed it was and these were swine that had been in contact with humans, and he showed for the first time that influenza was a viral infection, and presumably the 1918 epidemic obviously was due to an influenza virus of some sort. And in 1934, the influenza virus was initially isolated from human beings. But we had no viral preparations from anyone who died in 1918. People were long dead. No material was saved in a way that you could recover virus that would be active. I like to say live virus, but you know what I mean by live, right? And so this epidemic was essentially forgotten for all intents and purposes. Although it had severe impact over a short period of time on the global economy and on the U.S. economy, and it had social consequences, it really didn't, it just disappeared from view. Yeah, any questions? Okay. Let's see, how am I doing? All right. I'm on time, actually.
So if a woman is infected with a particular virus and then recovers and subsequently gives birth a few years later, does the baby have any immunity to that particular strain?
So the question is, if a woman is infected during her pregnancy, or even before the pregnancy, well, yes, she will have some of the mother's antibodies, but they won't last very long. They might last about 3 months, but then they'll disappear from the circulation because the baby himself, longer? 6 months? Okay, you like 6 months. He's a pediatrician, he knows more than I do. But yes, there is passage of maternal antibody to the fetus, which then, as the baby matures, dissipates, essentially. So, so 1918 was forgotten, and now we get to 1976. I hope you'll figure out why I'm jumping to 1976. But this is the pandemic that never was, and there's a lesson here that really resonates back to 1918. What happened is, in February of 76, there were 5 cases of influenza that occurred at Fort Dix, which is a recruitment center for initial training of Army recruits. And one of these 5 cases died. And as it turned out, after more investigation was done, about a total of 11 men who were at this Army base, where they basically aren't allowed to go anywhere during this training period, became ill. And it turned out that all 11 of these had a pretty severe case of flu. 1 person out of 11 died, which is maybe it's not statistically significant, but it was alarming. And as it turned out, after they did the studies of the antibodies that were in the bloodstream of 500 more of these Army recruits who didn't have any symptoms, they found out that, this strain, which is a brand new strain, and it came from a swine, it was a swine virus, an H1N1 virus, they had antibodies which suggested this thing actually had spread somewhat throughout Fort Dix. Now, this wasn't known immediately, but pretty quickly. There was a very rapid investigation by the people at Fort Dix and by several other government agencies, including the CDC. And as it turns out, which no one else knew at the time, that virus was never isolated again after March of 1976. On the other hand, right after these first few cases were observed, David Sencer, who was then the director of the CDC, met with his advisory group, the ACIP, which I was a member of. Were you, Phil? No, okay. And many other experts in flu to talk about, could this be the next pandemic that would have occurred after 1918? After 1968, sorry. Could this be the next pandemic? And Sencer made a decision based on talking with a lot of different people and on basically his own gut feeling about this. He recommended to the President of the United States, who was Gerald Ford, that there was a possibility that this strain of swine flu might become the next pandemic. Bear in mind, we didn't know, we thought actually 1918 was a swine virus, and it was called the swine flu in some places. But we didn't really know what it was because it was dead. And other people thought, maybe this could be as catastrophic as the virus was in 1918. It was possibly a swine virus in 1918. 10 times as many people died. And therefore, his recommendation to Ford was, we need to have a national vaccine campaign to try to protect the American public. And so President Ford elected to try to vaccinate 95% of the population. That was the goal, initial target. Well, it's a long story, and it was very difficult to get vaccine manufacturers to want to make vaccine when they weren't sure it would be worthwhile, it wouldn't pay well, there could be enormous liability if there were adverse events, et cetera. Congress eventually authorized essentially a tort reform bill that would indemnify the vaccine manufacturers, but they didn't really get any vaccine made until November. And then the vaccine campaign started. And as it turns out, some states did very well in vaccinating a large proportion of the population. Other states barely made a dent. Does this begin to sound familiar? And but in order to kind of get people to take the vaccine, Ford had himself vaccinated on television. I thought this was Dr. Salk, but it turns out it wasn't. It was someone else who gave him his shot on television. And actually, the reason he did this is because a couple of days before, 3 people who had gone to one clinic to get their shot dropped dead shortly thereafter. And the news reports picked this up and made a big hullabaloo about, well, maybe it was really the vaccine, and even one reporter noted that the vaccine wasn't safe. So in order to convince people that the vaccine was safe, Ford had himself vaccinated to show people he wasn't afraid of it. It would be perfectly safe. So a total of 40 million Americans were vaccinated against this virus. And as it turns out, about 1 in 100,000 plus people developed this syndrome, which had not been seen very much prior to that, called the Guillain-Barré syndrome, which is a kind of ascending paralysis. And of those, about 1 out of 1 million people died. So basically, because the fits and starts, and because now there'd really not been an isolate of this same strain in many months, 9 months, and because of the fear about the vaccine and the Guillain-Barré, which turned out to be associated with the vaccine, the whole campaign stopped in '76. The end result is it contributed probably to President Ford's losing the election, which occurred in November. David Sencer was fired by the new Secretary of HEW, Joseph Califano, and 2 years later, Professor Neustadt and Dr. Feinberg actually were asked to write a report for Califano about the swine flu affair. And in reading their report, which was written in '78, there are just so many warnings for what would happen the next time we tried to vaccinate the whole U.S. population. We have learned quite a bit since then, but some of the same problems still come up. And I think their conclusion was that Ford really didn't have any choice, and he wanted to err on what he felt was the safe side. But the issue about Guillain-Barré and the influenza vaccine has dogged people for all these years since '76, and has contributed significantly to some of the people who are very afraid of the vaccine. It doesn't account for the people who think the vaccines cause autism. They have other reasons for having their beliefs, but it has contributed to some of the unease about taking a new vaccine. And I would like to end up with some comments about that. So let's go back to 1918. So why should we worry about what happened in 1918 anyway? Well, first of all, there's something very funny about that virus, and if we knew more about why that was such a virulent virus, it would tell us a great deal of what to look for in the future and perhaps ways to prevent something like that from occurring again. So we didn't understand any of the virulence determinants. And indeed, another strain like the 1918 strain could certainly evolve. It happened once. It could certainly happen again. And as I said, if we knew more about the 1918 strain in particular, we might even have some novel therapeutic targets and some other ways to prevent infection in the first place in terms of our immunization programs. So, how are we going to resurrect a dead virus? Well, Jeffrey Taubenberger was a young pathologist who was at the Armed Forces Institute of Pathology, a now defunct organization, thanks to our wise Congress, which was a repository of a massive amount of pathological material from the armed forces and going back decades, including boxes and boxes and boxes of formalin-fixed tissue material taken from soldiers who died in the 1918 influenza pandemic. Now we carry forward to the modern era of sequencing and DNA technology and PCR amplification. And Taubenberger started in the mid-90s to see if he couldn't extract some pieces of the influenza virus RNA from fixed tissue materials and use PCR amplification to then get the sequence of the virus and essentially figure out what virus it was. Put all those pieces together and look at the huge database of information that's now known about influenza viruses and figure out what virus it really was. Well, so he started this, and he wrote an initial paper that appeared in a very prestigious journal, identifying the beginnings of the gene that encodes for the hemagglutinin of the 1918 virus. So he was actually able to get little bits and pieces of nucleic acid material out of some of these paraffin blocks. It had taken him and his colleague Ann Rich years to get this far. And now we come to Dr. Holton. This is how I got interested in flu. Johan Holton, who's shown in both pictures, in 1951 was a visiting medical student from Norway. And he went to the University of Iowa, I think as sort of a postdoc of sorts. And during his time there, and he was working in the laboratory, I think on some viruses, but during his time here, there was a visiting speaker who talked about the 1918 pandemic and what a mystery it was. And Holton, being a Norwegian, knew all about the permafrost, and that he realized that people who were buried in the permafrost could be still frozen. And he looked at the regions of the permafrost in the world, and then he cross-referenced that with the regions of the world where people had died of influenza. And I have to tell you that virtually every component of the world was affected by the 1918 pandemic, except for two tiny little remote islands. And he found this place called Brevig Mission, which is a Inuit village in northwestern Alaska that's in the region of the permafrost, and he decided he would try to go there, have access to some of the bodies that were buried in this village where virtually everyone had died of flu, and try to see if he couldn't resurrect the virus, the live virus, get it to grow in culture. So he went there basically on his own. I think the University of Iowa got him $1,000 to travel money. And he met the village elders and finally succeeded in getting their confidence and permission to exhume the grave. which is what he's standing in here on the picture on the left, and found some of the bodies indeed were still frozen, and he took some samples, but he didn't really have much of a way to transport these back to Iowa. He had sort of made a dry ice concoction, as I remember his story, and took the stuff back to the lab at the University of Iowa and tried to get the virus to grow, essentially, in tissue culture cells, completely unsuccessfully. So it was a bust. But he remained really intrigued by influenza, especially 1918, and he read Taubenberger's article in Nature magazine. And he thought, and I guess he realized from that article, and then he subsequently called Jeffrey Taubenberger, that Taubenberger's having an awful hard time of getting intact sequences, and he was just getting little bits of a few nucleotides and so forth, and that he really needed more material, and if he could get more fresh material, not formalin-fixed, paraffin-embedded material, he'd have a better chance of getting more of the viral sequences out. And so Holden said, I can go back to the same village, and I can, this time with the right materials and with some better support, I can get you some more material from frozen bodies. And so Dr. Holden went back in 1997 to the same village, again got permission to exhume the grave, and found one body that was still really relatively intact, not thawed, and took samples of her lung and brought them back to Taubenberger. Subsequently, Taubenberger and Reed and their colleagues have now determined the entire sequence of the 1918 virus, all 8 genes. And this is what they've done. They've taken the pathological specimens that they had and that Holton brought back. They've done PCR amplification, sequenced the genes, reconstructed them in the test tube, essentially built them again, put them into the influenza virus through a process called reverse genetics, looked at the contribution of each one of these 8 genes to the ability of the virus to cause disease in different models, including into tissue culture and also into a mouse model of influenza. And the bottom line is, I'm not going to show you the data, but the bottom line is that the entire 1918 virus is indeed about 10 times more lethal to mice than the background virus, and that several of the genes seem to be important. It's still not clear if there's a single gene. It probably isn't a single one of these genes, but we're getting a lot closer to understanding the 1918 virus. And we now know that it was not a swine virus. It has the typical signatures of a bird-like virus, but not any bird virus that has ever been isolated subsequently that we've ever sequenced. So it wasn't a swine flu. And in fact, the pigs that were having flu at the time of the 1918 pandemic among humans probably got it from the humans, not the other way around. And it was an H1N1. We know that because of not only his sequences, but you can actually tell when you look at specific antibodies from people, you can type the antibodies and you can see if they react with an H1 hemagglutinin or an H2 or an H3. So in fact, many years before, we knew that it was an H1N1 virus, but we didn't know what it looked like. And then you see the next pandemic was an H2N2, and it was much milder, and it had a much more typical effect on it. Mostly the mortality was in babies and in elderly people. And then in '68, we had another virus that emerged, H3N2, and that's what's been circulating for the last 40 years, predominantly. And again, a milder flu with about the same death rate as we normally see, again, in babies and elderly people. And then we get to 2009, where we have a swine H1N1, which is actually all of these viruses, to one extent or another, are actually derived from the 1918 virus. They have genes in common, or pieces of genes in common with the 1918 strain. But this is different enough that and of course we don't have anybody really, any significant number of people left over from 1918 to test how they would react to the new California virus. But I think it's substantially different. And I just put a few figures here. I'm going to at the very end talk about, what our actual experience is in California as well as in the United States with this latest pandemic flu virus. In 2005, when we thought the next pandemic might be the bird flu, H5N1, the CDC and the WHO started making estimates of the impact of something like the 1918 virus or something more like the 1958 virus on the number of illnesses and the number of deaths, how much hospitalization would be required, and so forth. And just look at what would have happened if it had been H5N1, which I think there are other reasons to believe it probably never will be. But we would have had 45 million visits to doctors' offices. This many millions of people would have been hospitalized. This many people would have been in ICUs. This many ventilators would have been required, which is, I don't know how many fold more than what we actually have. And we would have expected, and this is United States only, 2 million deaths. Now, a modest or a moderate flu like the 1958, if you just scale up the numbers for our current population size, we can anticipate 209,000, somewhere between 90 and 209,000 deaths, actually. And I think that our current swine flu, H1N1, is going to be like this. And that we're going to see a lot more people in the hospital needing a lot more intensive care than we've ever experienced before. And the last thing I read today was a report from the state of California that was just published today, showing that indeed we're ramping up and our hospitals are experiencing, and our primary care physicians are experiencing a massive onslaught of patients. So that brings me to the current time. And as you know, in the spring of this year, following our typical seasonal flu season, which was just winding down, there were these reports of this outbreak of influenza in Mexico and actually concomitantly in California. And in fact, the first isolate was actually derived from California, and so the new name of the virus is actually California H1N1, and it is a swine virus. But these are the early data that came from Mexico about who's affected by this new pandemic virus. And just the key point on this slide is that the median age of people is 20. Pretty young. And then if you look at the age groups where people were, now these are hospitalized patients, but it has a spooky similarity to 1918 in that people you wouldn't expect necessarily to be most at risk for acquiring infection are in these young adult age groups. Now, I want you to look at the right-hand part of the slide. This is what we know about the virus. Of course, now I think this virus was sequenced in a matter of days, maybe a day. So we knew immediately what the new strain looked like. And on the right, you can see that there are 4 different colors of genes. So there's one gene in this new pandemic virus that is a human This was from the old seasonal H3N2 virus. There are 2 yellow genes. These came from a bird in North America. There are 3 blue genes, which are from swine from North America, and then there are now 2 red genes. These are from Eurasian swine. So it's got genes from 4 different mammalian species. Now look on the left. This is a virus that was endemic in pigs in several locations in the United States, was causing swine influenza in several locations in the United States, and 11 people who were in close contact, almost all of whom were in very close contact with these pigs, came down with a moderately severe case of flu. Nobody died. This occurred in 2005. Nobody died. That was the end of the human cases. It was sort of like 1976. Nothing else happened in the human population. But now, when you look at the comparison between this virus, which was in swine, and a triple reassortant, in other words, it's got 3 different mammalian species of genes involved, and now you look at the new human one, this is a classic example of a reassortant virus that this virus got together with H3N2 and mixed up all their genes, and this is what came out. Now, we don't know where the swine are that might have had this combination. And it might have arisen in a human, actually, who also was infected with the swine virus. It's just not clear at all. There's no epidemiologic connection with pigs in either California or Mexico to show exactly how this occurred, but it obviously did. And so it does, again, exemplify this whole idea of why influenza can create pandemic strains by mixing and matching its genes once they get in the same cell. Now, this is the percent of visits to doctors with influenza-like illness. That's this axis. And this is the year and then the week of the year. That's how CDC represents the data. So in 2007, we had our peak of influenza, which started in 2006, in about February. And then influenza cases went down, or influenza-like illnesses. And then if you look at 2008, the peak of the season occurred a little bit later, like 1st of March. And then it went down. And then you get to the end of 2008, we started to see seasonal influenza, and it peaked here. Then all of a sudden we get this blip in April, and then it kind of went down, and now this is where we are today. This is probably a little spurious in that this is influenza-like illnesses, and if you remember, or maybe don't realize it, but when the whole news got out about what was going on in Mexico and stuff, our emergency room, for instance, at Stanford, was just flooded with people who were afraid they might have flu. And so a lot of the stuff that came through the door as influenza-like illnesses wasn't actually flu at all. It was other viruses. So I'm not sure that this is real, but one thing to note that is real is that even though it looks like over the summer things went down, this is still a whole lot higher number of visits than usual. And indeed, that's exactly what we observed here, and I think all around Northern California, is that flu continued to occur throughout the summer, and now we're really into full swing. In Region 9, which includes California, in the state of California, we haven't hit our peak of flu cases yet, and I don't know how high this is going to go. And this is just data from our own virology laboratory at Stanford hospital. And if you just look at the bottom for a minute, it tells you what kind of viruses we've been isolating from specimens. And the blue bars are the new strain of influenza A. And virtually all of the influenza that we get right now is all the new strain. There's a little bit of another virus called parainfluenza that's now in the community, and there's another virus called RSV that's now in the community. But for flu-like illness, it's all the new strain. And this is what's happening to our virology lab. The number of tests that are being ordered is also going up dramatically. And then finally, these are the data hot off the press from the CDC. And this is our region, 9. And the red line is 2009. And I think that's right. It's influenza visits, or influenza-like illness visits. And we're going this way. If you look, if I showed you the data from the East Coast of the United States, like Boston and Philadelphia and New York, they've had their peak already, and they're on the way down in terms of the number of cases. But we're just on the way up. And that's classic for influenza. It goes from the East Coast to the West Coast, and from the eastern part of the world to the western part of the world in a seasonal pattern. And there are, a lot of speculation about why is influenza seasonal. And in temperate climates, where the winters are cold, it's a time when people stay indoors. They're in more crowded places. The transmission is easier, and that's one explanation. There's more humidity in temperate climates, and that promotes the survival of the virus. But probably even bigger factor, I'm just guessing now, even in hot climates where there really isn't much of a seasonal temperature change, the winter months still, that's when influenza peaks. And what happens all around the world is that kids are in school in the winter months. And I think it's the fact that kids are in school, and kids are usually the vectors of contagion to the rest of us. I mean, the kids are the ones who are affected first. They have the least amount of immunity. They get everything that comes by, and they bring it home. And I think that's a big contributor to why it's a seasonal disease. The vaccine, as you know, is made in hen's eggs, and we have two kinds of vaccines that have been made for the last several years. The major one is an inactivated virus. That means although live virus is inoculated into the eggs, then when the material is pulled out of the eggs, the virus is inactivated with chemicals to essentially make it incapable of dividing and multiplying inside your cells. So all you're getting are really the proteins, the hemagglutinin and the neuraminidase proteins of the virus are what's used for the vaccine, and therefore you can't get the flu from the inactivated vaccine. You just make antibodies to it. There is a live virus preparation, and this is an attenuated virus. So they've taken the H1N1 strain and the seasonal flu strains, passed them through eggs to the point that they really have lost their ability to multiply very well. They do multiply inside the cells of the nasal mucosa for a few days, and they elicit an antibody response, but then they essentially stop because they're weak. And the whole idea of vaccination is actually to make antibodies to these two major proteins that are on the surface of the virus. Yeah. You have a question.
So, before airplanes, how did the flu move? Does it go back to birds? Does it go from a human to a bird, and then the birds are moving, and that's why it comes?
Sherry wants to know, before airplanes, how did influenza virus move around the globe? Probably not through the wild birds. It is coming through humans. Humans do travel. They've been traveling for hundreds of years. Yeah, but before airplanes, in 1918, we had ship troops, and we had people going on ocean liners. And it's true that wild birds carry the H5N1 to several regions of the world, and there was a concern about these migrating birds bringing the H5N1 virus directly to us. There was not any evidence that it ever reached the United States. It's a good question, but I think since it's a human-adapted virus, it really is people. It doesn't have an environmental reservoir. Yes?
Two questions. One is, how do you choose between the two vaccines? Which one is better? And the other one is, are there any plans to do vaccine preparation faster? Like you mentioned that technology is so old.
Right. Two great questions. Let me finish up with those questions. And I'm almost finished with my formal presentation, and so we've got plenty of time to answer really good questions like that. First of all, right now, we have a shortage, and and the reasons for this are entirely predictable. Our vaccine manufacturers had already started on making the seasonal flu vaccine at the time when this new virus emerged. So in order to even try to get people immunized this year, they committed to diverting their manufacturing plants to make the new vaccine. But they had to start late. Obviously, they couldn't start until later. And actually, they didn't start until August. And this is usually a 6-month process. So what's happened is that some of the manufacturers actually had to stop making the seasonal flu vaccine, and so we've actually run out of seasonal flu vaccine temporarily that we had started inoculating people against, and tried to, move over to the new strain, but it's going to take time. It is an old technology. And the distribution, it has always been a problem. Actually, our country, first of all, vaccine manufacturing generally is a market-based economic situation. Only 2 of the current manufacturers of flu vaccine are actually in the United States. There are 4 vaccines that are licensed by the United States, but 2 of the companies are actually, their manufacturing plants are outside the United States. And we don't have any control over those. Secondly, there's a huge risk involved in any company making a new vaccine, and especially with the specter of 1976 and Guillain-Barré. A lot of negotiation had to go forth by the NIH and the federal government with the vaccine manufacturers to induce them to make the new vaccine, and they all did. They're being subsidized by the federal government, which is, I think, entirely appropriate. But the next step, and the most critical step, which is the distribution of the vaccine from the manufacturers out to the public and out to the public health departments, has never been a streamlined thing. Typically, the vaccine manufacturers have middle people or middlemen who are the distributors of vaccine and sell their product to institutions, to doctors' offices, et cetera. They don't go through the state health departments. And if you might remember, when we were starting making plans for bioterrorism and stuff, it came to the attention, I hope, of the public that our state health departments are woefully understaffed, under-supported, and it's even worse now than it ever was because of the recession. So in Santa Clara County, for instance, we have had to lay off many of our public health nurses. We only have 3 MDs in the whole of Santa Clara County who are involved in our public health program. And and in our particular case, it looks like the communication between our counties' health departments and our state health department was not great. And so there were misunderstandings about how it would be distributed and how many doses people get. Anyway, we're on the short end of the stick here in California in terms of getting vaccine. So who should get I'll get all those questions. Who should get the vaccine that's available right this minute? I think you all know this already, But these are the priority people who should be first in the line. Pregnant women, they are at substantial risk for death if they acquire influenza, especially in the second and third trimesters. Healthcare workers with patient care duties. If you don't have anybody to take care of the sick people, that's a problem. Children and adults between the ages of 6 and 24 years. Adults in the same age, older adults with chronic medical conditions like asthma, diabetes, obesity, young children, and then people who have infants under the age of 6 months because we can't immunize those babies. Now, let me answer some of the questions that you have right now before I finish up. Yes?
Is it possible that somebody had H1N1 last Christmas?
Is it possible that anybody had H1N1 last Christmas? Not as far as we know. Yes?
Talking about the public policy on the previous slide, I think all of our vaccines are manufactured outside the United States, and a lot of foreign governments have said if it's a critical, life-threatening vaccine, we don't care, we're just seizing it and using it for our own people. So from your standpoint, sitting on the committees, what should we be doing as a nation to protect our own interests? Because if there is going to be an influenza that's going to have a high mortality rate, we're not going to get any doses.
Well, actually, you said all the manufacturers of flu vaccine are outside the United States. It's not true. Sanofi is in Pennsylvania, and one other vaccine manufacturer is in the States. Novartis is in Europe, and the fourth company is in Europe. I think what protects us right now, first of all, these are allies, if you will, and I think the negotiations that were carried out this year are exemplary in terms of understanding what the vaccine manufacturers needed to have in order to go forward in converting their facilities. And that is our best line of defense. There's no question about it. The vaccine is our best line of defense. There are a lot of reasons why many companies don't want to manufacture vaccines. It's not a big item. You know, it doesn't make much money, if any, and the liability is huge. And then you wanted to know if there's new technology that we're moving toward besides this old technology that's been around since the 40s. And the answer is yes. Several of the companies have the ability now to grow the viruses in animal cells, which is a whole lot easier, faster, and safer in terms of not getting it contaminated. And we're just not to the point of licensing by the FDA. And in order to use a vaccine in the United States, the FDA has to approve it. So those new vaccines will be coming down the pike. My friend who runs the vaccine program for Novartis thinks that within 2 years we'll have animal tissue culture cell-based vaccine. You have a question?
You said that the peak has already occurred on the East Coast.
Yeah.
Does that give you some kind of predictors for the situation on the West Coast?
Yeah, it means probably that we'll start to see some declines, I would guess, by December. But that doesn't mean it'll be gone. It just means that the largest number of infections will have occurred by then. But we'll still see H1N1 for some period of time. I think it's not really predictable about how long. There's actually flu around every week of every month of the year. It's just, it's a matter of numbers. I mean, people can get flu in the summer. As long as there are people who have influenza, they can transmit it to other individuals. It's just that the largest number of people with flu occurs in the winter months. And we also expect that the old viruses will also return. And we've had just a very few in Santa Clara County so far of the old seasonal viruses have shown up. It's just completely overwhelmed by the H1N1 right now. But we don't know what the mix will be, say, in April of next year in 2010. We are encouraging everybody to get vaccinated, both the old 3 strains that are in the seasonal flu vaccine as well as in the new strain as soon as you can, as soon as it's possible. Yes?
I wondered how long the immunity lasts, and also if there is kind of a genetic natural selection thing for families who survived the 1918 and were relatives of them, if there's kind of a, because they feel the plague is that way, that we have kind of some kind of genetic protection.
2 really good questions. One is there's some kind of genetic selection for, and could it be passed on to the descendants of 1918? And as far as we know, no. That there don't seem to be any specific genotypes of humans who seem to be more or less resistant. Now, how bad the flu is has a tremendous amount of variability, and that does depend on individual host factors, if you will. But as far as I know, there's no genetic basis for susceptibility to the flu. Because in a pandemic, at least 30% of the global population gets sick. That's essentially everybody is equally likely. And I'm sorry, what was your first question? Oh, how long the immunity lasts? Well, the immunity to the hemagglutinin and the neuraminidase probably lasts for several months to years, but because the virus keeps making these minor modifications over the course of time, it's really important, if you really want to have the most effective vaccine, to make a new vaccine. Make sure you've tuned up the vaccine to match the most recent strains. And it's probably not, those 2 proteins are not as the antibodies aren't as long-lived as one would like. The other thing that manufacturers are looking for is a universal vaccine. In other words, a component of influenza virus, not the hemagglutinin or the neuraminidase, but something that's stable so that we could immunize people once. And that might last years and years and years. So far, magic bullet hasn't been found, though. Yes?
Along those same lines, I'm wondering if there's any studies or thoughts about the fact of having, say, a child, like 10 years old, that gets vaccinated, and then gets vaccinated every year for their entire life. They could have, 70 vaccinations of different slight variations of the flu. Is there any thought of what impact that might have?
So what if you start getting vaccinated when the time, say, starting at age 2, and now you get vaccinated every year for the next 60 years? Would that have any negative or positive impact on you? And the answer, I think, is no. Our bodies are programmed to be able to recognize, I don't know, was it 1 million? At least 1 million different proteins or antigens. And so looking at 60 over the course of your lifetime is a drop in the bucket. It certainly doesn't use up any of your immune system to do that. And that's one of the reasons why I think the concern that some parents have, for instance, about their babies or their kids having to get, as many as 37 shots for childhood diseases, and worrying about whether that puts a big burden on their immune system to see that many different vaccines, for their body to see that many vaccines is really not a problem at all for the immune system. It's tuned to be able to deal with millions, essentially, of different antigens. And in fact, in the course of just eating things and being exposed to what's around us, we're being constantly challenged by a myriad of different antigens all the time. Yes?
If a college student got the swine flu in September, should they still get vaccinated? Because this tweaking or what they got in September is with the vaccine? Or
Yeah, it depends on if you're very sure that they got the new H1N1, which is highly likely if they had documented influenza. Now, there are other things that look like influenza, Certainly, it wouldn't hurt to get vaccinated. I would say if there's a shortage of vaccine and there are other kids at college who haven't been able to get vaccinated, then I'd kind of step aside of the line and come in a little bit later. But it certainly wouldn't hurt to get vaccinated, and you can't be absolutely certain unless specific tests were done, which in fact mostly weren't done by September because we just assumed virtually every influenza-like illness or every flu illness was the new strain. Yes, you wanted me to ask someone? Oh, one more on this side.
You referred to the business that's been referred to at different age groups, whether it's 55 or 65, something about older people not getting it. And I saw about 2 weeks ago, I thought it was on the CDC site, saying that this was probably because of exposure to 1957 pandemic. But how could that be if that's an H2N3 or whatever? It's not an H1N1.
Good question. It is an exposure to 1957 because, as you're right, it was an H2N2 virus. It is true that people over the age of 60, probably 65 for sure, seem to have a level of antibody that reacts with the new strain. And if we look at the curves of who's getting infected, many fewer elderly individuals are getting infected than we would anticipate. So they are more protected. And it's not clear at all what they would have seen 60 years ago. It's just a hypothesis that they have been exposed, and I don't think it was the 1976 vaccine campaign. That was a swine H1N1. At least I haven't seen any recent reports, and I just checked today to see if there's anything more about the 76 vaccine, but I think it's still too early. I mean, it had to have occurred 60 years ago for these people to be protected. And we don't know what virus it was. And it's possible it's something, one of the seasonal flu viruses or whatever that was used, but I think it's not known. One point I do want to make, though, is even though older people are relatively protected compared to younger people, if they get flu, they're much more likely to die. So there's a much higher mortality rate when adjusted for the number of people in that age group of people who've gotten flu who are going to die. And primarily that's because older people are much more likely to have other comorbid conditions like diabetes, heart disease, lung disease, asthma, etc., than young children. So the kids are having the highest hospitalization rate, but they actually have a very low death rate. Pregnant women, on the other hand, are not any more susceptible to getting infection, but they are much greater risk for death and complications. So I think we've used up our time, and I'll stick around if anybody wants to talk with me later. Thanks a lot.
So in thanking again Dr. Tompkins, this was a talk just looking around the room that I think really riveted everyone's attention for obvious reasons, and I think it's hopefully one that both informed you and I think to some degree reassured you as well. I want to just let you know, in case you wanted to continue with us as time goes on, we are going to be posting the winter session of this course on November 5th, and registration needs to be completed by November 30th. So starts November 30th. So if you want to come back, and I can tell you the program in January and beyond looks fantastic, you're welcome to do it. And But till then, we'll see you next week. Next week, we're going to be talking about the environment and risk for certain infections as well. So see you then.
For more, please visit us at stanford.edu.